Everybody, welcome to another Friday night Bigfoot episode. Tonight we have a special guest, Dr. Haskell Hart, and to talk about the Sasquatch Genome Project, a failed DNA study, his new book. Well, we're going to get right into this. We got Dr. Haskell Hart with us right now, and Dr. Haskell Hart, welcome back. It's the second time in 4 years that we've had you on here. Since the last time you recently got a new book out, and a lot of people are already familiar with this book. We're going to get into talking some minor details, and Dr. Haskell Hart, I'm going to give the floor to you. How about you? There's a lot of people that may not be familiar with you. A lot of people do really know you. So how about you take it away from there? Tell the audience who you are and where you come from, and give us a little bit of background if you don't mind.
Sure. Well, I have a PhD in chemistry, and I taught for 10 and a half years at the University of North Carolina in Wilmington, physical and general chemistry. And then I got a job with Shell in Houston as an analytical chemist and research manager, which I worked through 1981 and then retired. And since then, I've kind of been doing what I want to do. And one of the things that came up in 2013, of course, that you're all aware of, is the Sasquatch Genome Project, Melba Ketchum and others, 9 other co-authors. And I've been a naturalist and an outdoor enthusiast, a nature student all my life since I was 5 years old, actually. And so this piqued my interest. I had heard of the Patterson-Gimlin film, etc. And I hadn't really studied the matter very much, but I thought with a DNA study coming out and being a chemist, and by the way, DNA is a chemical molecule. It's actually 2 molecules wrapped around one another. So I thought it'd be a good opportunity to learn a little bit more about DNA, which I had already known about for 50 years, hadn't done much with. And I thought this would be a great way to prove the existence of something that has been a mystery. And I was really optimistic. I was not expecting to spend 7 years on this. I thought, I'd learn about how to do database searches. I would hopefully confirm what the study found, and that would be the end of it. But it didn't turn out that way. The more I got into it, the more inconsistencies and errors I found in the paper. And I had to educate myself along the way about searching databases and what is a good hit and what is not a good hit, or so forth. And of course, I didn't work on this full time. I had other things in my life. And so I did publish a blog, bigfootclaims.blogspot.com, and I put my findings out there. I had a lot of hits, but no criticism, a couple of ad hominem things, but nothing scientific. And I thought, well, gosh, it's 7 years now. I've kind of wrapped this up. There's not a lot more that I'm likely to find about it. And so maybe I need to put all this together into a book that people can buy online. An ebook as well as a paperback. And that's my case, and they can study it and come to their own conclusions. And I wrote it so that with enough background in the book that people they don't have to be scientists. They don't need to be geneticists. They should be able to understand it. They're going to have to, pay a little attention to the early chapters. And I thought, well, I want this to be my expression of what I think the truth is about this. And I don't care if I won't make any money on this book. There's not enough people to, to pitch it to, I don't think, in the Bigfoot community. I wanted to get it out because it's a statement of what I think is the truth, and I think it's sorely lacking. There are some highfalutin geneticists, and they had armchair opinions on this, but they never did any detailed comparisons or to the Ketchum work or any independent searches of the databases. And I think you need to do that if you're going to criticize something. You can't just read over it. Some only read the introduction of the abstract and the conclusion and said that this can't be. That's not good enough. And so that's why I did this. And I hope that enough people who are interested will read it. And I'm very, very open to criticism and comments. I mean, nobody's got all the answers. I may have overlooked something. I'd like to have that pointed out. And so I'm hoping that, programs like yours might inspire some of that. We'll see.
Yeah. The problem is there's two problems with people in the Bigfoot world actually reading the entire study. And the first problem is, as far as I know, still to this day, she's charging $30 to read it. Now, I'm sure other places they can be found, but on her publishing site, her self-published site, I believe it's still being charged for, isn't it?
No, it is. There is a new website. Well, it's not new anymore, but I paid my $30 because I had to hear about this right away. But since then, there's a website called SasquatchGenomeProject.org, and you can download the paper for free, download all the supplementary data, and download the nuclear DNA sequences, the mitochondrial DNA sequences, and that's what I base my work on. I had bought this, but you can get it for free now. SasquatchGenomeProject.org.
Mm-hmm. Yeah.
And tell Melba that I sent you.
Yeah. And also the other problem is it's just true is that a lot of people, including myself, so I don't exclude myself from this. A lot of people have a hard time interpreting that kind of nomenclature, that kind of data. I mean, some people could be taught to understand, sequencing, but just to going cold, even a reasonably intelligent person to going cold, like it might not be their thing. They might have more of an artist brain or a math brain, or
Quite honestly, when I first read the paper, I didn't understand much of it either. And I've got a lot of degrees and stuff. It's not my field. My field was physical, analytical, and inorganic chemistry, not biochemistry. But I have enough background in chemistry that I was able to learn the relevant biochemistry of it. But I have to admit that for the average person, they would not understand that paper. They may understand the abstract and the conclusion, and then they decide based on emotion whether they agree or not, and they haven't read the details. I've read that paper or parts of it dozens of times actually in doing my work, and that's what it took. Now, a geneticist like Todd Disotell or others could read through it and understand it, but even some of them said it didn't make sense to them.
Yeah,
so,
and
I appreciate that. This is not something that if you haven't got a background in science, you're going to pick up right like that. I've tried to write a book where I can lead you into it, and I may or may not have been successful in that. I tried. So don't feel bad. Most of the people in the world are just like you. And, this is not something you're born knowing about.
Yes.
Now I'm excuse me. I'm interested
I like to
know.
make mention too.
Yeah, I'm sorry. I'm interested to know.
Oh, go ahead, Brandon.
A little bit personally, Haskell. Did you have an experience with Sasquatch or Bigfoot or anything that got you interested? I know you said that you were a bit of an outdoorsman, but
Yeah.
what got you interested in it originally?
Well, I've always been interested in natural history. I pressed wildflowers in my Chicago 4-inch thick phone book and so on since I was 5 years old. I was aware of the Patterson-Gimlin film. I didn't know what to think of it, and I kind of kept up with some news articles between then and now. But it was the DNA aspect that got me interested in the Ketchum study, and I thought this was something I could really get into based on my chemical background. And as far as Sasquatch, yes, I have had a sighting. It was in chapter 19. You can read all about it. It happened in southeast Oklahoma, in the Ouachita National Forest, which has been known to produce a lot of sightings. And it was a clear morning. I wasn't under any influence. It was in bright sunlight, which is very unusual for sightings, surrounded by nice greenery, lit up, and a orange-red Sasquatch. And I only saw it for a couple of seconds, but I know what I saw. And so I believe it exists. I was on the borderline before that because science, you at least want to see something before you say you believe in it. And so you can read about that too. And it was a wonderful experience. And I was very lucky because this was early on. I've been out there before that and after, and I've never seen anything. I did hear what I would call a definite primate whoop on another occasion, and it goes something like this. Just like some hawking afterward, similar to some of the sounds on Sierra sounds.
Okay.
Sierra, you know what I went, Ron Moorhead.
Yeah.
But it was a whoop. There's no animal in those woods
yeah.
that makes a noise like that. It's a primate whoop. And so I had those two things, plus another, what they call the Ohio howl in Louisiana. Where I was camping on Caddo Lake. And, that's a long howl. I don't need to repeat it. You know what it is. So, I've got some evidence that I think it exists, and I'm looking for more. I want to get some better DNA samples, and I'll be out soon after the election, and we'll see if I can come up with something more convincing or not. One thing I'm hoping, and by the way, this was an early version of this picture you're showing of my book that has a wrong word on it. Forward should be FOREWORD. There's two forwards, and I picked the wrong one, and everybody's making a big deal of it on this cover. Lately, it's been corrected, but nobody's perfect, And then, of course, don't judge a book by its cover.
Well, that's exactly why I brought this up here, because I am aware that in a post, I won't mention who reshared the post about your book. We know they're not from the United States. However, I saw that you were under a little bit of attack, and a lot of other people had some input because they were judging a book by its cover because they're seeing what the images that you used with the red cross mark with Patty on the cover. And that's one thing I thought we could get cleared up right away because a lot of people were in our passing judgment because they are judging a book by its cover. The title should speak for itself, You're not denying that Patty was real or not real. I think people should understand to take consideration of the title itself, and then do what I did: buy the book, get the chance to read it, and learn what it's about. Because there's some people, I'm sure, that are not familiar with the original study with Mel Buchanan and the Sasquatch Genome Project and everybody that was involved. And even to this day, there's people defending it. Till this day, Melba Ketchum has a following, more of a cult, because they're learning what the information that there is hearsay, instead of understanding the facts and knowing the details between the proper study, the sequencing, and everything, how everything goes in order, and how things were basically incomplete in the original study. So, but yeah, I think
Yeah.
people, I'm just sharing this little bit of information that they need to understand, that you cannot judge a book by its cover.
Yeah, and
I got a few small books that I put out there. I, but yeah, I just wanted to share that. But go ahead.
well, oh yes, I've seen a lot of the posts on this. It's called kind of a little uproar in one of the other Facebook pages. This is my first book. I'm 77 years old. I designed the cover to save money. And my message there was that, according to this book's findings, the Sasquatch genome study did not find Bigfoot. It did find a bear and a dog, which have these little green check marks on their pictures. They're smaller, so some people may not have noticed them. It was not in any way intended to say that the Patterson-Gimlin film is a fake, or that Patty is not real, because actually I've read about it, and people who studied the film, especially Bill Munns, and I am convinced that it's a real animal. It's not a costume. A costume could not have created those muscle movements and so forth in this picture at that time. They had no spandex, they had no Lycrex or whatever that other stuff that sticks to your skin is, and I'm convinced it's authentic. Maybe I got too cute with the cover, but people need to get over it. And they don't have to buy the book. They can look at Kindle. They can look at the Kindle version at Amazon, and they can read the first few chapters. That includes the preface, the foreword by Jeff Meldrum, the introduction, and part of chapter 2.
Mm-hmm.
And a lot of my motivation is explained in there. And so they don't need to buy the book to get a little feel for what's involved. And certainly, if you read that, you will find that this is not dumping on the Patterson-Gimlin film as Iger Burtsa thinks I did. So that's all I can say. Maybe I shouldn't have tried to be quite so cute. I didn't think it was all that cute, really. Maybe the picture's too big, smaller, but I'm designing a cover, and I'm sorry people feel that way. I think really most of them are not going to like the message in the book, and it's just convenient to dismiss it all by criticizing the cover. That's very easy to do. You don't have to educate yourself by reading the book. And if you've got this hard emotional feeling about it, it's a good way to just dismiss it. That's my opinion. I won't say that's true of everybody, but I think a lot of them that have criticized it, they won't go beyond the cover because they don't want to hear the message. Don't ask questions you don't want to hear the answer to, said my former director at Shell.
Yeah.
And that's what I think about it.
Right. A little bit of cognitive dissonance going on there.
Yeah.
So I do want to ask you, since since we brought it up now with the forward, so Dr. Meldrum wrote the forward for your book.
Yes.
And I saw, I saw Dr. Meldrum little over a year ago. Let me get some noise. Little over a year ago, and during his speech, he kind of just he kind of destroyed the Ketchum study in about, I don't know, 2 minutes. Which so again, like, as somebody who can't dive into the minutia of the science, it's always good to hear a qualified person sort of boil it down for you.
Yeah. Sure.
And so, so I look forward to reading your book, and of course seeing, the actual forward from Meldrum. Yeah, I thought that was interesting. Now, did you just did you know Dr. Meldrum beforehand, or did you just reach out to him and ask him, like, say, hey, I've got this book, you know?
It goes back a few years.
Mhm.
I attempted to publish my first paper, and I wanted to get his opinion of it before I went ahead with it in an online journal. And he's a busy man. He gets tons of email from people. They want to have their samples analyzed, this, that, and the other. And somehow it got lost. And 2 years later, after he had read, 2 or 3 years later, after he had read my blogs, he said, well, would you write an article for the Relic Hominoid Inquiry, which is, his online journal? Are you familiar with it? At Idaho State?
Mhm.
Relic Hominoid Inquiry, you can look it up.
Yeah.
Yep.
And I responded, and he said, oh my God, I looked up and I've got your email from X years ago, and I never opened it. And he was very apologetic. And I said, no problem. I've learned some more things since, and I will write you an article. So there is an article in the RHI, Relic Hominoid Inquiry, the title is DNA, the potential in relic hominoid inquiries, something like that. And I review the previous work of a number of people who've looked at samples that were thought to be Bigfoot but weren't. And I gave some recommendations on DNA analyses and database searches. And so that is out there as of 2016. It's a bit old by now. And so that's how I got to know Jeff, really. It's hard to get his attention, but he wanted a paper and he got it.
Mhm.
And we've been friends, and of course, occasionally we exchange some email and so on. I did write a book review of Scott Carpenter's book,
Right.
called Truth Denied, the Sasquatch Genome Study.
Mm-hmm.
I made Jeff aware of that. And then I told him, I think I need to write my own book on this. And it took me, I started about a year ago, and I'd still be writing if it weren't for the virus, which kept me kind of contained and with a lot less outdoor activity and stuff.
Yeah.
And so after I finished, I thought, well, maybe he'll return the favor. He asked me for a paper, I'll ask him for a forward. And he agreed enthusiastically. He wrote it. I'm also a good friend of Ken Gerhard's, and I reviewed his book and commented on it before he published it, the last one being something like Sasquatch Field Guide or Sasquatch something like that. It's his last book. And so Ken wrote me a little short paragraph of endorsement. And so that's how Jeff and I have gotten to know one another.
Okay. Do you I'm curious, do you touch on Scott Carpenter's book, defending the Ketchum study. Do you touch on that in your own book?
Yes.
That we're talking about? Okay.
Yes. One of his quotations that I have right up front, I think in the preface, as a motivation was he says that to this date, no critical scientific studies have ever been made of the Sasquatch Genome Project. And that, later on in his book, of course, he says it's all a conspiracy, that mainstream science is trying to hide this somehow and defame Melba Ketchum. I got better things to do. I didn't even know Melba Ketchum before this. I don't hate her. I hate Hitler and Stalin and Mao and people like that.
You and me both, buddy.
Yeah, not Melba Ketchum. I mean, I've got an opinion of her, but I try to keep it on a low key. So anyway, Scott, there are a number of places where I quote him in the book, and sometimes just use the information that he provided, which is useful, and sometimes rebut it. And the same with Melba Ketchum. There are a number of quotations there and exchanges between us that I think people need to know about.
Exchanges. Explain.
Emails. I reached out to her. Listen, I wanted to know what she was thinking about this, and when I found some inconsistencies and some errors, I pointed it out. I tried, and the more I got into it, the less friendly she was with hearing about it. At first, I thought that, well, she had found this, but I had to learn the hard way that a 95% match with a database on some genes that are conserved, which means they're carried on from one animal to another in evolution because they work. A 95% match is not a species match. It's got to be 99 or more. And just a 4% difference makes all the difference between whether you really have a match or not. And she didn't realize this, and I didn't realize it at first either. You're not born knowing this. You got to try some stuff. You got to do some comparisons, and learn about it. So the more I got into it, and the more comments I made about it, the less friendly she was. And so we haven't communicated in a few years.
We're not surprised.
Her last comment on DTIFA, which is a Swiss journalist's blog, which covers some Bigfoot things. He interviewed me and then interviewed her on that, and then I got to rebut it. And so there's a complete thing. It's in chapter 18 or so. And her last comment about me is that I'm a paid government agent and a quack.
Oh, awesome. Hey,
yeah, I'm still waiting for the paycheck.
Wow.
Yeah, well, where can I sign up for that?
Yes, I guess maybe it was at $2,400 they sent me and my wife for COVID.
Yeah,
I don't know.
Yeah, but
other than that, I've never gotten a government payment.
Right.
And I would never accept any such thing.
Right. Because I get exactly this much money for coming on here and giving my opinions about how I view the world in reality versus delusion.
Yes.
And are you aware? I'm sure you're aware, Haskell, that she's a veterinarian basically.
That's correct.
And not a genetic scientist.
Oh yeah, I know that she took a 5-week course in Kentucky, as she said on one of her radio interviews, to learn about how to use DNA in diagnostics in animal cases, like which dog bit my horse's ass. You know what I mean? That kind of stuff. And she went to court with things like that. But she's I guess she'd call herself a forensic scientist, and at some level she is. But now there are questions coming up. Am I to answer these?
Sure. Yeah, answer.
Okay. Here's a good one. Here's a very good one.
Yeah, this is what I wanted to address. Yeah, 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 Darby Oka. He's from North Carolina State University.
Yeah.
He's actually, very brown. He was actually one of my speakers at my
Oh,
one of my Bigfoot conferences here in Virginia. So yeah, he's got this question right here for you. If you as you see it, yeah, Dr.
right.
Hart, did you find anything that you found redeemable in the Ketchum study?
A very good question, and thank you,
And I'll
Darby.
let you take away that.
Yes, I began to look at the different mitochondrial DNA sequences, which were provided in the paper. That's the DNA in your mitochondria that comes from your mother. Only your mother can transfer it, and it only gets transferred to the next generation from another female. I only have brothers, but if I had a sister, she could transfer it to the next generation. My brothers can't, and I can't. We all have our mother's DNA. It's only 15,568 bases compared. That's 15,000 compared to 3.3 billion nuclear DNA bases. And I began comparing her samples, and I found that 4 of them had some common mutations, which were extra in the sense that they are not involved in that particular haplogroup, mitochondrial combination of mutations. They're extra, but they're very rare, and they're not found together in any human mitochondrial DNA that's been published in the database. And not only that, there's 4 of them, and not only even 2 are found together, and some are found singly, but very rarely. So I thought, well, let's see, could this have relationship to any apes? And so I took all the families of apes, and I compared these DNA sequences to them, and I found that except for chimps, the other apes and monkeys and prosimians, that's like lemurs and so on, they all had much more occurrence of these mutations, which I don't know what to make of it, because I think it takes a pretty good geneticist to go further with this, but at least it's something unusual about these 4 samples. I'll tell you where they were. One of them was the famous Physiological. It had 2 of these unusual mutations.
Mm-hmm.
One of them came from New Mexico. Another one came from California. And another one came from British Columbia. So I think these samples have to be related in some way, genetically. And I went back and looked at her raw data. She does publish some mitochondrial chromatograms of all the bases. Actually, they're called electropherograms to be specific. And I checked, and these seem to be present in sample 26 anyway. So they weren't an error. And so I think there's something for somebody else who has a laboratory and the ability to analyze DNA samples. I don't have a laboratory. I only analyzed her results, her sequences, GT A
Now,
C,
is it possible that those anomalies could show up in the bear species?
They didn't in the bears that I looked at, which are the polar bear, the black bear, and the brown bear, and the panda. No.
Because I'm sure you're aware that only because you cited that one of the samples in what you were just discussing came from Justin Smeha.
Yeah,
And
which is a bear sample.
he also gave a sample from the same, which was an inconclusive piece of flesh that he found later. He didn't know. He thought maybe this could be related or
Right.
not.
There's no direct connection to what he shot.
Right.
It was 5 weeks later under 2 feet of snow,
Exactly.
searching around with his dog.
Right.
And
But he also gave a sample of that to Brian Sykes
yes,
to determine that it was in fact bear. So
it was a black bear.
yeah.
And here's the thing: that black bear sample, the nuclear DNA is certainly black bear, and the mitochondrial, because you used human primers, you would only pick up impurities. You wouldn't even pick up the black bear, even though it was the main sample, the main species in the sample. The way they do these human mitochondrial things, I sent from friends of mine who are mentioned in the book, a dog, a cat, and a horse DNA swab from their cheeks into family tree DNA, the same company that Melba used for her mitochondria. And the answer they came back in each case was did not amplify, which means that their methodology is geared to human DNA. So they use certain primers to initiate this replication, and those are human, and they won't work on animals. I proved that. And so you could have a bear sample with just a trace. You touch the bear fur, and you send that in, and they dissolve it all up, and their primers won't pick out the bear. They'll only pick out human because that's all they're looking for. These companies, they do your family tree. They don't deal with dogs and bears and other animals. So all the methodology is only going to pick up humans. And if humans are the contaminant, that's what you're going to get. And she was so, I don't know what, naive, I guess. She thought that was her real sample. But,
but
it's still interesting in that.
Yeah.
What if this bear, let's just, they have fantasy football. I want to have fantasy DNA. Here's what could have happened. The bear, 2 bears fought over the Sasquatch carcass. And in so doing, contaminated themselves with Sasquatch DNA. And in fact, the independent study by Tyler Huggins showed DNA that actually was from 2 bears. It had extra peaks in the microsatellite look. So, and bears contact one another, just like people. You and I shook hands. Well, they take some DNA. They're going to have both of ours, And so here's 2 bears fighting over a Sasquatch DNA sample, smearing it all over them. And one of them, with his claws, rips out a piece out of the other guy's, a piece of flesh out of the other bear, And that it just falls to the ground.
That sounds like a pretty likely theory, yeah.
Well, I don't know if it's likely, but it's a way that I can explain some of this, that the human part in the mitochondrial was not quite human. It's got some extra mutations. Maybe it is Sasquatch, and maybe Sasquatch is nearly human. I'm beginning to think it may be. And that way, depending on what analysis you use and primers and so on, you could see bear or you could see this human-like Sasquatch, perhaps. So that's the way I come out on this. And of course, much, much more data needs to be collected from legitimate samples, hopefully from, a Sasquatch that you see deliver the sample in hair or something to a tree, or leave some blood, or you shoot it, or whatever. That's what we need, but it's wishful thinking at this point. I don't know.
Yeah, I believe the hair actually is really not such a good source of DNA, is my understanding.
Well, here's the thing. Human and primate hairs have very little medulla, which is the inner part that has the DNA. And so, and you cannot do nuclear DNA. There's not enough DNA. You have to just do the mitochondrial because it's 1,000 to 10,000 times more prevalent in the sample than the nuclear, because there's more mitochondria and each one has more DNA samples or sequences. And so, but there's a paper out by respectable people who did Brian Sykes analysis, and they took some old forensic samples where the cases are dismissed and they don't have any names associated. Some of these were decades old, and they were able to sequence mitochondrial DNA from less than half a centimeter of hair without the root. The root has a lot more DNA,
Right.
but just in the mid section of the hair or the tip, half a centimeter, that's not much. But if you do have some medulla there, and the medulla in primates is not contiguous. It breaks up, and there's gaps, and then there's a little, or there may be none in a sample. If you have a little bit of it, these techniques are so sensitive now that you can amplify that and you can sequence it. They did it. And so it is possible from just a trace of DNA to get a sequence, mitochondrial sequence.
Yeah.
It's been done. And I think that's what happened with a lot of kitchen samples. They were trace contaminants.
Yeah.
I can't believe you go out in the woods,
Yeah.
collect 111 hair samples, if you could find that many. And then they all come out to be human or human-like.
Well, sure, because
That's not likely. There, there's a few hikers. There may be a Sasquatch or two, but most of them are going to be other things. I think deer, rodents, foxes, coyotes, bears, whatever. There's way more of those than there are Sasquatch, or we'd have gotten some better pictures by now, right? So
yeah,
I don't believe her when
a whole different subject with getting pictures of them.
She said they did both a hypervariable sequence, which is a part of the mitochondrial, and cytochrome B, which is another part, on all 111 samples, and they all came out to be 100% human. I don't believe it. I want to see the sequences. I don't think she could afford to do that. It would have cost over $300,000, I think. And she never published the sequences. I mean, a statement that dramatic needs some raw data or some results to prove it, I think. Okay, Tina Carson. I thought Melba used 3 different labs. Also, she had blood and tissue samples from police and hair from other reputable researchers. I just started watching a few minutes ago. Sorry, I'm like, yes, she, well, first of all, she had samples from all over. Some of them were ex-policemen. Some of them, Scott Carpenter contributed a whole lot of samples, other Bigfoot researchers. So they came from all over. And as far as labs, yes, she used about 3 different labs to sequence, and another research organization in Texas at a university to interpret the results. And so, yeah, that's pretty much correct. But none of these labs had experience in forensic wildlife identification. In other words, these labs do police work on human beings. All their methods are geared to, did you or did I leave
Yes.
DNA at this crime site.
For
They're not geared to what kind of animal is this. That sounds like a trivial difference. It's not. The whole methodology has to be different when you don't know what you're dealing with. And they didn't do that. And I put a lot of fault on this guy, Fan Zhang from North Texas Institute for Biological Science or something. He did the analyses of the DNA results and the interpretation and coming up with human when it was bear and dog. He really blew it big time. I tried to communicate with him before I published. I wanted to hear, did I miss something? What do you think? Here's what I think about what you did. No answer. Stonewalled. In fact, none of the other co-authors except the microscopist responded. And he said about his work on the microscopy, I only take pictures. I don't comment on the paranormal. And I had asked him, how could the DNA pictures that he took show single strands and doubles? It's supposed to be double-stranded, but there were regions of single strand. And I asked him, is this what kind of degradation, microbial, chemical, sunlight, whatever? And he didn't want to answer that simple question. And the most logical thing is that those samples were degraded by microbes, and microbes chew up one of the two helices and leave the other one, or sometimes they chew them both up if you wait a while. So those pictures are very typical of degraded DNA. And when you get degraded DNA, your results are going to be very, very unreliable. You get sequences of things that were not what you were looking for because they were not primed. They self-prime. I explain this in one of my chapters. And in fact, the mitochondrial DNA data that you put on our webpage, I bought the software to look at it and look at all these peaks, ATGC peaks. And two of the samples had bear in it, bear nuclear DNA in a so-called human mitochondrial sample. And most of these were unusable. They had so many overlapping peaks, you could really not do a sequence on most of them. So I only got like a third of the whole genome sequence from her data. And that third did not agree with her published sequence. So it's not reproducible. It's contaminated. It's degraded. It's obvious, but you have to read the details. It's all in the details.
I got a question. Yeah, I got a question for you if you don't mind. In your scientific opinion, what is it going to take, short of producing a body, for science to accept that this is a genuine species, that this thing is out there? I mean, pictures aren't working, video aren't working. It seems like anytime there's DNA presented, all it does is cause an argument. So is there anything other than a body that's going to force science to accept this?
Maybe not, but here's what I would try to do with what resources I have is get a good enough DNA sample that we can sequence a decent, large enough 300 or 400 base segment of mitochondrial DNA and show that it is a primate, but not a human.
So what would be a good source of DNA? The DNA you're speaking of, what would be a good source for someone to gather?
Well, something like flesh or blood is the best.
Right.
It has the most concentration of cells. Hair will do if you have a decent sample and it's got a medulla. Some do, some don't have the medulla. That'll give you mitochondrial. If you get enough blood, you could actually do some nuclear DNA, which would be even more convincing. Microbes, there's no body there, but yet they're recognized as species, microbes, and a lot of it is based on DNA. Viruses, there's no body, but yet we seem to know that we have CV19, based on its RNA in this case, I believe, sequences, which is related to DNA. So it might be possible. Of course, accompanied by pictures would be a lot better. And one possibility I'm trying to work through and propose something to somebody who has resources in the lab is water samples. Now, are you familiar with the work that was done on the Loch Ness recently, where they collected water samples up and down, left and right, different depths?
Oh yeah.
A group in New
I'm
Zealand
not.
at Otago University in New Zealand.
Yeah,
And
they came up with a. Yeah. Didn't they find results of
Dozens of
eels? Some kind of
species.
eel?
Yeah,
I remember
that's right.
that. Yeah.
The closest thing it could be is a giant eel. But there's a lot of eels in that lake, and a lot of them are just small, and so, but sometimes eels grow to great lakes, great distances, size. And they found birds, water birds, and land birds where the DNA washed into the lake, and mammals washed into the lake, and reptiles and amphibians and fish, of course. It's full of different kinds of fish. And they were able to sort out all these species. They haven't published the final report yet. I'm really waiting to read it and learn about it. But I think you could do that with streams. Like there's an area in, like I mentioned, in southeast Oklahoma, that has a lot of streams that come together. And if you could get these samples and get some DNA, you might find all kinds of interesting animal species, but you just might find this rare primate as well. Still, I think in order to be put in the textbooks, if you want to use that as a criterion, you're probably going to have to get a body part, maybe not a whole body, preferably a head or a hand or an arm or a leg. Because most species have a holotype, somewhere in a museum somewhere, there's a bird that they say is the gold standard cardinal, and everything else has to be measured against that. Or a rat or a badger or whatever. Somewhere there's a holotype that you have to measure against if you want to say you have the same species. And so probably a body part is going to be required. But you can get a good up on it and find out where they exist if you do some of this environmental DNA work that I mentioned.
True.
Or get another sample off of a tree or something.
Yeah, I mean, environmental DNA, though, the problem with that is that it requires professionals and money. Two things that don't exist in the Bigfoot world.
Right. Money, and what else?
You're a professional. Like Meldrum's a professional. We've had professionals in the Bigfoot world, but the vast majority of the Bigfoot world are broke and not professionals in science.
Yeah.
So
but they can collect samples, perhaps.
I'd love to see more environmental DNA done, but it has to be done by professionals.
Right. You can't do it in your garage. No way.
Yeah, not researchers.
Even though I've thought about it. My wife wants our car parked. She really liked both cars parked in our 1 3/4 car garage. And there's only one now and the rest is full of junk. So it's not going to happen there. But you're quite right about that. You need a lab that does this on a regular basis. There are commercial labs. And I think some of them may be better than a university because they're being paid to do it. And if they don't do it right, they don't get paid. The university can say, oh, well, it didn't work out, or I don't have time for it. My students want to do their research and the instruments aren't available, this and that. So I'm looking into it, and I'd be willing to support this financially, at least to a degree, if I can get some reliable person to do the analyses. Because you might find some other interesting things. This area that I'm talking about, they've got recordings. A group went out there and they think the red wolf exists there, which is a thought to be near extinct or extinct species that's hybridized with coyotes and therefore, hybridized itself out of existence. But they had some recordings of howls that aren't coyotes and they're yet dog-like. So, you might find something like that would be valuable.
Yeah.
Plus all the other animals, that. So I'm looking into it. Believe me, I think it has some potential.
Yeah, but again, it requires money and professionals.
Right.
So
somebody's got to pay for it. Yeah.
Somehow you have to get those people interested, especially if you're looking for what we're talking about with any kind of primate connection that's not completely human, but some kind of unidentified hominid, I guess you could say.
Yeah.
And so the thing is, how do you get these professional? It's like a chicken and the egg thing, man.
Yeah.
The Bigfoot world is so full of crap to most scientific professionals that, I mean, we can have a objective scientific discussion here, but we're like a small portion of the Bigfoot world. You know what I mean? So scientists don't have anything to do with it. So then people always go back to, well, we have to kill one to get science involved. And it's like, it's a never ending, like chasing your tail situation.
Yeah, it is. It tends to be that way. Unfortunately, I think we need some lucky breakthrough, and I don't know. I would love to see it. I don't expect that I'll be the one to make it, really. There are others that are more involved out in the field. I think that's what it's going to take. And but if we could get water samples that showed an unknown primate, that would focus the area. And unfortunately, a lot of guys and gals are going to come in there and probably scare them all away. So I don't know. You have proof that they're there, and then they're not there because you advertised it.
Yeah, they have a funny way of
one thing
that script on us. Sometimes they come in and scare us away.
Yeah,
that's been in it tens of thousands of times. So yeah, go ahead, Daniel.
Just one thing I wanted to make mention too. Now, one of the gentlemen that commented or had asked you a question earlier, Mr. Darby Orcutt from North Carolina State University.
Yeah.
One of the things he shared with me in the past, and I think you may find that you may have heard of this. He was actually involved with a kind of a project that was kept on the down low, kind of private, and it involved the late Dr. John Bindernagel and a couple other primatologists and biologists. And Dr. John Bindernagel. Yeah. Well, this project, one of the things involved with was as far as collecting DNA and proper ways to collect samples that might get science to consider, just the possibility. Different protocol steps. For example, one of the things that were mentioned when people find tracks, they take samples of the track. Both the track itself and away from the track in the same soil, especially for mainly, doing chemistry lab studies to see if they could extract, perhaps DNA out of that. Have you ever heard of anything like that as being like something likely to, that would be a good work?
In fact, I copied some papers off the internet, scientific papers. There was a study in Alaska, and they took soil samples from a cliff at different levels, meaning different time and periods in time going down. And they were able to find mammoth DNA, rabbits, moose, wild horse, and I think I got most of them. There were 4 or 5 species, I think, of all extinct animals, or some were extinct and some were close to what we have presently. And they got this from up to 10,000-year-old soil. Think of it. This stuff's been sitting in the soil for 10,000 years, and they were able to get it out and get enough DNA to prove what they had. I've also got a paper from a group that did cave studies, and they got DNA from cave lion. And I think cave bear has done the same, although they have bones of cave bears, and they're extinct, at least 10,000 years. So this is possible. Yeah. And boy, if you can get a bone, that's much better than soil, of course. So
yeah,
there's possibilities. Now, I'm very disappointed that the Olympic project, those nests that they found, do you recall a couple of years ago, that looked like might have been Sasquatch nests with woven branches and twigs and stuff that you wouldn't expect rodents or other animals to be able to do. And so far, they haven't found anything in the soil or however they collected samples there. They would be environmental samples as well, most likely. I don't think they found any hairs, but it hasn't been reported. I wish they'd report it, even if the results are not Sasquatch, just to see what's possible and how the methodology works and whether it might apply. The methodology might have been good. There's just wasn't a Sasquatch sitting there, although nobody seems to can think of any other animal that could have made these. They're too big for rodents, and bears don't weave stuff and so on. But I'd like to see the results. I'm going to hit on Jeff Meldon to cough it up. It doesn't have to be successful.
Absolutely.
The methodology can be successful.
Oh, yeah.
And it can lead to other studies where the samples and the conditions and environment are appropriate. So anyway, I was disappointed, but that's the way it is sometimes.
Yeah, there was an image of Cliff Barackman from Finding Bigfoot. I remember they came across what appeared to be a large nest on the ground, and it clearly looked like a nest.
Yeah.
But then again, there was the picture that I saw had Cliff. Yeah, it had the picture of Cliff laying inside of it. Of course, there's your there's your contamination right there.
Yeah, I'd say collect your samples first, then take the picture. But you can look around. There may be some hairs, or the thing pisses in the ground. That's good. Collect the soil. And there's a lot of opportunities, and maybe somebody will find the right nest sometime. There's a lot of convincing looking pictures, but that's it, just a picture.
And yeah, you would think there would be just all kind of hair samples inside those nests too.
You really would, yeah. I would think you could find hair. So I don't know what they found. They need to report it. I think it would help us all, lead us in the right direction, maybe.
Yeah.
Yeah, and like you said, it doesn't have to be successful. It doesn't have to have Sasquatch, DNA in it. Regardless of what it is, like you said, yeah, it's a learning experience. It'll give us intel. It'll give us some feedback, regardless.
Yes, and if the methodology.
Now, as far as you were talking about
yeah, go ahead.
Yeah.
The water.
Now I was just gonna add on and make mention about the water. Yeah, is there anything, like, for example, me, Wayne, or Pat, if we go out during our hikes or whatever out in the woods, and we decided to take water samples from the stream. Is there anything simple that we could do to get that tested, or is that something you would still have to send off to a lab? Or I mean,
Yeah,
would a
you're gonna
lab
have to
even
send it.
bother
You're gonna
testing
have to
anything like
send
that?
it to a lab, and you need to get in touch with them in advance, and of course agree to who's paying for what, and they need to provide sampling equipment, and you need to do it so you don't contaminate it. And one simple way is just to drop a bottle and let it fill up with water and put a cap on it. But there are other systems that pump the water through a filter, and you can get much more greater volume of water through that way. And then you take the filter and extract the DNA off of it and analyze that. And I think that might have more promise. You could let it sit there for a while and collect a lot more sample than in a liter. Typically, they do about a liter of water. You could pass gallons and gallons through it and collect the DNA on the filter. So you need to work. Don't just go out and collect water samples. I don't think anybody would want to spend any money on that. There's too many uncertainties. But you got to work with your lab analyst.
Yeah.
And that's what I'm trying to get into and see if there's a reasonable place at a reasonable cost that's willing to help me work through the sample collection. And, I think there's some promise.
Yeah.
Now this might be a stupid question. I don't mean, and I'm sorry to interrupt you, Pat. This might be a stupid question, but what are you looking for with water DNA? I mean, what exactly is water DNA? Is it like saliva, or like from bathing or walking through it?
What happened? What it is cells that slough off, like a fish swims there, but cells slough off of its gills. Or you take a piss in the water. Or you go swimming and the cells slough off your skin. Or an animal on land rubs up against something and cells fall to the ground and get washed into a stream and washed into the lake or whatever, wherever you collect it. So it's sloughed off cells, basically, that have the DNA in them. So it can come from anywhere. A snake slithering across the ground, or a water snake, or a fish you're going to get a lot of fish in water because they're the most abundant thing in that water. And you're going to get more DNA from them, I think, than anything. So, yeah.
Again, that kind of thing, environmental DNA, especially from a stream, that requires professionals who are dedicated.
Absolutely.
And know what they're doing.
Nobody has average people. I can include myself in that. We don't have sequencing equipment. It takes an instrument in a lab. It costs tens of thousands of dollars to even deal with the DNA once you get it extracted, and that you have to extract it properly too. Not contaminated, concentrated, and you need to have the right machine, the right primers, yada yada. And now some of this is done by what they call it's kind of a generic term, next generation sequencing. What you'll find when you do this is a lot of these cells have released their DNA into the water, and you'll get short segments because they're decomposed. And what you have to do with a computer is look for where these sequences overlap, and then you've got two of them stitched together. Okay? Where the sequences are the same, you can assume that these two match in a linear sense. And then you look at the ends of those for something that aligns with those. And you stitch together these short sequences. And that's quite a job. You need a really good computer. You can't do it on your PC. You can only they use what they call either supercomputers or a bunch of linked servers to be doing it all at once. And it's a big computational thing. Just think of, we have 3.3 billion bases in our nuclear DNA. Imagine it broke up into segments that are less than 100 bases long and stitching that back together by the overlaps. Because some DNA will break here, some will break in other places, different molecules in different places. So there's some overlap. And you definitely need a professional with the right software and the right computer.
Yeah.
And, I am not that person. Sorry, please don't send me anything.
And nobody in the Bigfoot world is either. I can.
No, I mean we come from other backgrounds. You know, I've gotten into the computer side of it, but I don't have a laboratory. I can't do that. I can understand some of it, but I need to go through a lab with somebody to be really proficient and talk to them in advance. Don't collect samples till you've got a plan for how you're going to collect them, how you're going to transport them and preserve them so they don't decompose on the way in their hot back of your pickup truck. You need to put them in alcohol or freeze them or yada yada and get them there, and then what they're going to do afterward. You have to understand that, or you're not going to get what you thought you were. I'm afraid to say.
Yeah.
Unless you're very lucky.
No, what we need is something. So let's just say not a body. What we need is something to titillate the scientific community a little better.
Yeah. Something better. Yeah.
Yeah, and just something to show them, to get them more interested.
Right.
So if we had hours and hours of Patterson-Gimlin type footage, not 30 seconds.
Yeah.
Obviously, I think that would go a long way.
Yeah.
Things like that.
Or even stills, really good stills, a series could help. But also some DNA studies.
Yeah, it'd have to be a whole lot of stills.
Even an environmental DNA study that showed an unknown primate and met all the criteria for a DNA study to the experts. It would lead them to believe that this is worth pursuing. Yeah, that's right.
That would whet the palate of more people. The problem is getting to that initial point. We don't really have anybody in the Bigfoot world that can even conduct these environmental studies. I would think there are probably some people, but that would require funding.
Yeah. How
do you get the funding? Who's paying for it? Like, again,
right.
Chicken and the egg, dog chasing his tail. There's got to be something else in between to whet the palate of let's say even just 50 scientists, like to get them interested. And obviously the Ketchum study was not that thing,
Unfortunately not. Actually, a half a dozen really good scientists that are willing. See, it's hard to get a grant for something like this to support. Your laboratory there's primers you have to buy. They're expensive chemicals. There's other supplies, yada, yada. It's hard to get a grant for something like this. So somebody is going to have to go out on a limb and say, well, I'm going to take it from this grant and I hope it works. Or get somebody to fund it, or donations, GoFundMe, I don't know, and go just actually do the work. And that's not easy because these guys have other agendas. They're publishing papers. They've got graduate students that need to write a thesis, and they could be working on this for 10 years and not have enough for a thesis. It just might turn out that way, and that's not fair to the student. So they got other. Yeah. Sorry. Go ahead.
Yeah, Haskell. I just saw this comment pop up, and I was wondering if you wanted to respond to this right here.
Yeah. Well,
It's
I don't think it's very convincing if they're talking about the Ketchum study.
They have to.
It's not convincing. It was thought to be DNA of Bigfoot, but I'm
Yeah.
convinced it is. Except possibly some of these mitochondrial samples that have unusual mutations. They might actually be Bigfoot. There's some mitochondrials that might, but it needs a lot more work. You can't draw any conclusions from her results. They're not complete enough. And some other people have tried and they've got some interesting results, but nothing convincing. Not nearly convincing.
Exactly. I mean, that's kind of why we're here tonight, and why we're talking about your book, because my God, that study came out in 2013. You know, it has been thoroughly debunked.
I think so, but not everybody's convinced.
Sure, and I'll shed some light on that if you'll let me. You know, look,
Sure.
there's a lot of people who are looking for answers, and there's a lot of cherished beliefs. Okay, and the whole Melba Ketchum study became this weird, almost flat Earth thing. That's what it is now. Like if you believe that the word of the Ketchum study is gold, and you're going to spout it as, well, we know what they are because it's already been done. Then I mean, you're a flat earther. Like if you just buy into that, then you're no better than the people who think the Earth is flat, because it's just a cherished belief you have.
Yeah.
And it was all built on, the whole thing was sort of designed, I think. There's a weird connection. Or have you heard the term, angel DNA?
Yes, I've heard it.
And your understanding of that
Yeah.
is what?
Well, I try to deal in real things. The word angel itself is not something that science can deal with, really.
Right.
Angel, one way out of all her dilemma, or her, when she's backed in the corner, is say, well, these are not worldly species. They come from outer space, and therefore anything that we do to study their DNA is inappropriate and won't give good results. I've heard that a lot from some of her followers. And so, I can't deal with that. The people are free to believe what they want, of course, but
Well,
somebody gives me a sample they say is angel DNA, I'm going to hand it right back to them.
well, are you aware of people like Scott Carpenter and David Blightus, and others that were involved way back in 2013? Are you aware, that there's become a very clear attachment? It's not coincidence that all these people that defend the Ketchum study are all fundamental Christians.
Well, I won't say they all are. She's got thousands of followers on Facebook, and
True.
I'm not about to say they all are fundamental.
I'm talking about the people who were close to her. That would be
maybe so. I don't know them well. I don't know what Scott Carpenter's beliefs are, for example. It's never come out, and I really, it's not anything I'm curious about, really. There's that, but I don't know that that's a big factor in any of this. There's some interesting questions coming up. I missed the previous one. It was a pretty good question. History Channel had Bigfoot DNA. Well, I'll tell you, the History Channel, there's been some studies and a little bit of DNA from that Snellgrove Lake thing with the nails in Ontario. It was sent to a professor at the University of Minnesota, and he found one extra mutation that's common with chimpanzees. And he didn't say how long the sequence was, but that's not convincing. You and I could have a rare extra mutation that might just happen to match a chimpanzee or something. I think that's what she's referring to, the Snellgrove Lake thing.
It sounds familiar, yeah.
Yeah, it was up there in Ontario, and the cabin had been destroyed and raided inside, and so they thought they'd catch this by putting a board with screws stuck up through it, sticking out the top, right by the door. So the next time somebody came in, they'd
up
leave
in Alaska.
blood. Yeah, it was Ontario though, and maybe they did in Alaska too, but this was Ontario, and
okay.
they did get samples of the blood, and Scott Disotell had trouble with it. There was something interfering with the amplification, and they sent it to, I think his name is Kurt Nelson at the University of Minnesota, and he was able to somehow get around that interference and get a sequence, but with one mutation, you can't prove anything. I mean, it's tempting to say, oh, it must be ape-like because there's one that's like a chimp, but that can be just random. We all have some of these extra mutations, and I'm sorry and I think Nelson appropriately said it wasn't, convincing or definitive, but it's a start. It shows that you could do this actually, and maybe the next thing that steps on it might be a real Sasquatch. Maybe that one was. We don't know.
Yeah.
Could have been a human.
It's just TV. I mean, at the end of the day, man, it's just TV.
Yeah.
It's not a published journal.
And unfortunately, the sample stayed out in the wilderness in the wild there for some time before it was collected. And that always raises questions of contamination and degradation. So it wasn't a fresh sample by any means. But of course, DNA can be obtained from quite old things too. So, I'm not dismissing it. I think it's great that somebody did that. And we just need more of it somehow, somewhere.
So, Daniel, can I please clear something up? I was I misspoke.
Yeah, go ahead. Yeah.
And this is why, just so everybody understands, man. When I brought up Scott Carpenter and David Pilates, and that they were sort of PR people for. Well, Scott Carpenter contributed to the study.
Yes.
And at the time, Pilates was he was kind of a PR guy, and these guys are known like Christian fundamentalists, and the only reason why I bring that up is because there's a very like clear narrative to me that is which has come out recently in the past 10 months, is that there was this narrative trying to be built that they were Nephilim.
Yeah.
Biblical.
Scott Carpenter wrote a book about it, The Nephilim Among Us. Yeah.
So I'm being completely fair when I say.
Oh, it's true.
Yeah, that he's a Christian fundamentalist, which there's nothing wrong with that. I'm not saying there's anything wrong with that, but there's this connection to try and create a narrative that they are Nephilim. And I think that it's my opinion that the entire Ketchum deal was built around that narrative to try and like push that narrative forward.
I think it came later. I think when she ran out of excuses and she got everybody with any credibility was shown this to be a bad study with wrong results. I think then she started to drift to this supernatural. Because, you know, you can say if it's supernatural, then no analysis on Earth is going to reveal the truth about it, because it's so totally different from any animal on Earth that your analysis is inappropriate. It's just an easy way out, I think.
It is.
I don't know their religious belief. Well, I know Melba is pretty devout Christian. I don't know if she's evangelical or what, but
well, see, I have looked into their beliefs, and
yeah.
That's what you'll find. And it's not a coincidence at all to me.
Mm-hmm.
Not a coincidence to me that it is, mother, human, female mother, unknown, origin.
Yeah. Mm-hmm.
And that is the Nephilim narrative.
Yeah.
Fallen angels who laid with the daughters of men.
Yeah.
And that's not a coincidence to me. And also at the time, even before the study was released, the fallen angel, not fallen angel, but the angel DNA was somehow leaked. That wordage was leaked. And then, David Plitus breathlessly went on air on Coast to Coast to say, "Oh, no, I assure you that never happened," to defend it. And then we have 10 months ago, Steve Istel kind of bringing it all back up with his big following on YouTube. And the whole entire thing from 2013 came back into the Bigfoot world for the first time, really. Great.
Well, you know, you're right about Scott Carpenter. He wrote a book, The Nephilim Among Us. So it's obviously, I haven't read the book, but I will say from the title, it sounds like he believes in them. Otherwise, the title is inappropriate. But so you're right about him, and I think Melba has drifted that way. I think, you know, she didn't get scientific support from real scientists, and so now she's drifting into the supernatural. That's my.
Well, I mean, she basically self-published.
Yeah, and I don't criticize that, because what did I do with my book? I mean, I don't have.
You're not publishing a scientific theory. You know how that works.
It's science. But I did self-publish it because I don't think any publisher, first of all, they cost a fortune to publish a book.
Well, of course.
And I don't think they could get enough of a market to justify it based on their business model. And it's easy enough to do, and I thought I would do that and get it out there. That's what Scott Carpenter did, and Melba's got some self-published books. They're the fantasies, you know.
No.
Bigfoot.
Yeah, mine.
Fairy tales.
Yeah.
Yeah. But she had a.
Yeah, the few books I got are self-published.
She had an amazing. Yeah. She had a pretty significant scientific claim.
Oh yeah.
Proof that Bigfoot exists. You don't self-publish that. You get it published in an actual scientific journal.
And she tried, and they rejected it twice.
Yeah.
Nature, which is a premier journal of all areas of science, they suggested some changes. She really didn't make the major ones, and then revised it, and they rejected the revision. And then this other journal I've never heard of, the Journal of something zoological, something or other, they rejected it, so she bought it, changed the name, and published it, you know. And really, that doesn't impress me, but I was willing to pay my $30 and look into it and see if it was a valid study and had some conclusions that could be supported with results and data that looked good. And, you know, I did not hold that against her initially. It doesn't sure doesn't look like the way to get something accepted by mainstream science, but I took it for face value. I really did.
Yeah, and I commend you for that. I do, and I look forward to reading your book.
Well,
Definitely.
great. And please ask me any questions. You know how to get up with me now. If you got questions, I'll try to explain things or answer them. And I hope people will take enough interest to learn something. You know, they may not understand every single thing in there, but even something is worth it, I think, you know.
Oh yeah, definitely. Oh, don't worry. I'm gonna I will definitely contact you via Facebook, and
Great.
yeah,
Yeah.
I'll tell. I'm not gonna say it here because I say it all too often, but I'll fill you in on how we move the ball down.
Yeah, and you know, nobody's got all the answers. Nobody's got the final
No,
word on this yet that I know of. So, you know, I'm open to criticism, and
no.
that's the way science advances, really.
Yeah.
And I'm part of it, and I don't want any special consideration. I
No.
certainly don't want a bunch of people just saying, yeah,
Yeah.
just
Right,
because
yeah.
they don't like Mel Ketchum or whatever. Give me a good reason, and that's much better.
Oh yeah, but no, it's like as far as the phenomenon itself, you know, the Bigfoot phenomenon, you know, yeah, I'll give you my opinions on how we
Good.
move the ball down the field
Sure.
towards science. You
And
know,
it may, if you share them with others, it may encourage them to do likewise. You know.
yes. Well, I figured out how to observe them, so
Mm-hmm.
yeah, I'll fill you in on that.
Yeah. Yeah.
Okay. Well, I've enjoyed it.
Yeah. One thing that was
Go ahead.
brought up. Yeah, people keep talking, and I see it just got brought up again. Just the word Nephilim and the angel DNA thing. You know, I
Yeah.
know a lot of people have their own beliefs, and you know, and I usually try not to pull religion or the Bible into this, but you know, and
Mhm.
people may have their own interpretation of what things mean, and in the Bible,
Yeah,
I believe that comes with a lot of not reading the rest of the scripture. It's like, again, it's like judging a book by its cover. But when you start reading one piece of scripture where they get the Nephilim is, and understand what the Nephilim is, the fallen angels, and understand what fallen angels, what they actually are, where they went to, you know, without reading the scriptures, they're getting the one mindset of, okay, fallen angels came to earth, the Nephilim were the blah blah blah. Nephilim is just giants. There were human giants back in those days. The fallen angels, when they fell from heaven, they went to hell. You know, and yes, demons do exist. You know, where they do roam the earth. You know, blah blah blah. But in my honest belief in the scriptures, they have nothing to do with humans. Because if you read the scriptures, angels do not give. They're not given in marriage, or no. They do not have a relations as us humans do. You know, with other humans. You know, that's that. You know, everybody has their own belief, but that's my standpoint on the Bible. I was debating if I was going to share any of that because like I said, I try to stay away from all that. You know, with
Yeah.
all the Bigfoot talk, but a lot of people like to keep bringing that up. You know,
but
but it's something that's I guess misinterpreted. So
well,
Well,
there
a
is.
lot of
but
opportunity for that.
that's my
Yeah.
2 cents. I don't.
It
And
is.
you're right about just taking a short passage from the
Yeah.
Bible and trying to run with that. It always needs to be put in context, and you know that's a whole other area. I try to avoid getting involved with it. And it's possible the Bible is very metaphorical in places. It's possible these so-called Nephilim were actually some kind of Bigfoot or something. And that's the only way they could explain it, is that they fell from heaven or whatever, you know, because they didn't think they could have been of natural origin.
Right.
So you know.
Yeah. Well, here's the thing about the word Nephilim is from the Hebrew Bible,
Mm-hmm.
and
Yeah.
and so and then it gets interpreted into the King James, but even theologians, like hardcore theologians to this day, will disagree with each other about what interpretations
Sure.
of that in translations, where something was lost in translation. So like theologians that have nothing to do with the Bigfoot world disagree with each other
Yeah.
about the interpretation of Nephilim and fallen angel.
That's right. Some things are difficult to translate, and different scholars have different translations of them,
Yeah.
especially from an ancient language like Hebrew. Um, Another example, the Moses taking the people across the Red Sea and so on. There's passages there that you can interpret different ways. Supposedly, the Pharaoh's name was Shishak in one version. And depending on how you translate the hieroglyphics, it could be Ramses, because the symbol for the Esh is some wheat blowing in a field, I think. So, there's possibilities of different translations, interpretations, and I'm trying to deal with real samples and so on. So
Yeah, exactly.
it's interesting though, because the Bible has a lot of actual history in it. Archaeologists have proven a lot of the places and people that are mentioned in the Bible, and so there's some useful information.
Oh yeah, even found evidence.
Scientists. Yeah, Mount Sinai.
Yeah, they found evidence of the ark.
Yeah, well, they think that's another
yeah.
debatable thing, but there were floods. Actually, the Black Sea used to be a smaller freshwater lake, and at some point, and it's thousands of years ago, the Mediterranean breached the sort of natural dike between the Bosporus and what's now the Black Sea, and salt water went into it and raised the level, but the salt water was denser, and it did not mix very well with the fresh water. And so there's a layer of salt water that's still down below there. And it's been great for archaeology because it killed all the freshwater worms and things that eat wood. And so there are ships there that are thousands of years old that are preserved in this saltwater environment down at the bottom of the lake. So you could call that a flood. Something caused the Mediterranean to rise and spill over. It's very interesting. And finally,
yeah. Here's a question right here, and
yeah, go ahead. Oh, is it?
yeah, right. I believe it's referring to Melba's journal.
Yeah.
If I'm
right. Well, there's
reading this right,
that the editors conspired against her. I don't believe this. This is a Scott Carpenter claim and a Melba claim that the editors either purposefully or delayed her publication, hoping that Sykes would come out with his beforehand, that he was involved in this. I don't think editors of a journal of that reputation would do that. I think they're looking for new science, new good science. And I think they probably did their best to give it a fair look. And quite honestly, one of my chapters is on these peer reviews from Nature. And believe it or not, I did not know that they were leaked until a bit afterward. And I had come to almost every single conclusion and criticism those four Nature reviewers had, I discovered myself and agree with. They're very sound criticisms. And she says it's all bias and prejudice, and she never really answered the key questions very well. She always had a red herring or a sideline, like, for example, it couldn't be a bear because none of the laboratories we sent samples to had ever analyzed a bear before. So she's assuming that the thought is that it's a contamination or something, and that's not it. The sample is a bear, and it's just a red herring. Okay, if you read, there are, oh gosh, could you put that one up again? There are real
Oh,
flaws in her
yeah, I'm
work,
sorry.
her paper. It takes no conspiracy to reject flawed work. I agree. Now, Curious Cryptid is a recent friend of mine. He reviewed my book and he checked my database searches, came to the same conclusions, and he wrote a very short commendation at the beginning of the book alongside with Ken Gerhardt. So he and I have been over this in great detail with a lot of emails, and he has part a lot of check work, which I greatly appreciate because that takes time. And so just to let you know, to be fair, he's a believer in my work. Does Dr. Hart believe that science?
Yeah, here's another question right here.
I don't think so.
Yeah. Do you believe
I
that the
really
scientific
can't
conspiracy?
believe that.
No.
No, I don't think there's some conspiracy here either to deny the existence or to prove it. Either one. People can be wrong, but I don't think there's an ulterior motive. I don't know. People say that samples skeletons sent to the Smithsonian have disappeared. Now, here's a good one.
Oh yeah, I'm sorry.
And agree fully. It's very flawed and now being used to push
Yeah, some of the. Yeah,
Yeah, I think the concern is that people who might have believed in Bigfoot, after they understood the Ketchum study, they saw that that was flawed and therefore incorrectly conclude that it doesn't exist. I mean, that's the only thing you conclude is her samples were not properly analyzed or interpreted. Okay,
Yeah.
but it says nothing about the greater issue.
Yeah. The truth is that the Bigfoot community was very let down by the whole thing because
Yeah, a lot of people were.
it was built up. It was built up into this thing that they could believe in or like hope in, right? Like, oh wow, they were made to believe that it was legitimate or whatever, So all this other stuff kind of came out after.
Well, I'm guilty. I was really enthusiastic. I thought this would finally put the whole matter to rest, and I was disappointed. I mean, but I was interested enough to try to figure out what went wrong and so on.
Yeah.
And,
Well, see, then you know exactly what I'm talking about. People were they were let down by it.
Yeah. Some more than me because they had a bigger stake. They'd been out in the woods for years, which I hadn't been at that point. And, they had seen things and made casts and this and that. And they were hoping this would validate some of what they found,
Yeah.
But,
so if the scientific community says, this is a bunch of BS, and the Bigfoot community goes, this is a bunch of BS, there's no conspiracy.
I don't think so. I can't get in the minds of the Nature's reviewers. I just don't think they're that kind of people. They would not have gotten so far in their careers to be editors of Nature if they were that kind of people. I don't think,
Yeah.
And I can't prove it. I mean, I don't know them personally, but it's just very unlikely that they would be up to some of this. I don't know. It's not a national security issue that has to be kept under the cover or anything like that, like discovering some new source of energy or, it's natural history, whether it exists or not. It's not going to change our national security or anything like that. So I don't see a government conspiracy at all. And
Yeah.
I'm still waiting for my first paycheck
Right. Yeah.
if she's right.
I mean, I'm sure you're aware of the Flat Earth Society, right?
Yeah, yeah.
Well, what's one of their biggest claims in the Flat Earth Society is that, because they have to address it this way, is that NASA are liars.
Yeah.
NASA itself, the NASA entity is a big giant lie, and that's a conspiracy to cover up the fact that the Earth is flat by projecting narrative that the Earth is round. Yeah. It's the same kind of delusional thinking. It's
disparaging government agencies and so on. I mean, that goes on all the time, especially this election year and so on. I just think they got better things to do than try to hide the existence of Bigfoot. I don't see how that gains anybody anything,
I agree.
Maybe I'm naive, but I don't see it.
I mean, love him or hate him, if Donald Trump knew about Bigfoot, you think he could keep his mouth shut about it? Hell no. No way.
Well, I have to say that any president, not just Donald Trump, does not know everything that's going on in the government at the time.
I agree.
So, regardless of what he thinks, there could be other things going on. So I just don't think it's likely. I would like to know where all these supposedly large skeletons are that were sent to the Smithsonian. That would, yeah.
I think that's just ridiculous.
I don't know that it ever happened. There are pictures, but pictures can be faked.
Well, I mean, look, you're a doctor. Like, you have scientific knowledge. You understand how gravity works. And I mean, what are these skeletons? How big are they supposed to be? What are these skeletons?
Well, you'll see pictures of them from time to time on the internet. Some are 8 feet tall and so on, found in the Middle West. Some of them dug up from some of these mounds in the Middle West, in Ohio, Indiana, Illinois. Supposedly sent to the Smithsonian and can't be found now.
Yeah.
But, I don't know what to think. The cataloging system could be faulty, or they disappeared. Somebody stole them, or
some of them are supposed to be like 30 feet tall.
Oh, really? Yeah. That's
that's not physically possible.
Yeah, I don't think so either. They'd have trouble walking around, and that's
trouble breathing.
Yeah.
Their circulatory system wouldn't work properly.
You wouldn't think so, not
primate form.
Yeah.
Yeah.
The animals that are that big are all marine mammals that float and get some buoyancy,
Yeah.
Whales, in other words, whales. 30 feet is not realistic. 8 is possible.
Yeah.
There's some new stuff coming out. I don't know how true it is, but they're finding somewhere in, where is it? Is it in Venezuela, where they're finding large skeletons like that buried 8 feet or so, 7 or 8 feet. We'll see. I don't have anything definitive. It's just sort of casual news without a lot of details. But be interesting if it's true.
Of course.
Well, so where are we? We about to wind it up? Have we covered it? And of course, if you want to do another session at any time, let me know. I'm not sure what's left to discuss, but that's up to you. I'm sorry I wasn't able to respond quickly enough to all these questions. Some of them were statements. Here's one. Well, this one relates to something else that I think I should answer first. I will answer this one too, but somebody asked, was there anything good to come out of the Ketchum study? And I think I've answered that in the sense that there were some unusual primate-like mutations in some of the samples that might indicate a primate origin other than human. So, and the most bad examples of Ketchum study, I don't think they understood how to use all the databases that are available through the National Center for Biotechnology Information. Not all the sequences are in the same database, and unfortunately, the one they used had very little black bear data. No polar bear data, which is the next closest bear. I had quite a bit of panda data because panda and polar bear are studied the most because they're threatened, you know. So
Right.
people want to understand the population dynamics and so on. So there's a lot more sequence of data available for them. But even so, you do a search and everything comes up 95% matching, all different animals that are totally unrelated. And their conclusion was that it must be unknown because nothing matches and there's no clear preference for one type of animal over another.
Right.
Actually, you can't match something that's not in the database. There just wasn't enough black bear data over those same regions of the DNA genome as that were measured. Now, recently, 2 years ago, a whole black bear nuclear genome was published and put into the database.
Right.
So that's the whole nuclear genome. And I was thrilled because I've been looking for this for years, for 5 years before, as a matter of fact. It finally came out, and when I searched their sample 26 against this complete nuclear genome, boy, I got 100% matches for a lot of regions of the genome and over large regions too, like over 1,000 bases. And so, that kind of that's one of my chapters, about 15, I think. But before that, there was other data, RNA data, which is related to DNA. Some of that matched black bear very well. So, and then there was data that wasn't published in the in the government database that was published outside that I searched, and I got very good results there too. So you have to look around a little bit. They just searched the most common NCBI database, and unfortunately, it wasn't complete, and so they drew the wrong conclusions. That's their biggest mistake. I could name some others, but let's see. The creature most unlike us in DNA is the octopus, and we are around 85% like them. That's interesting, and Curious Cryptid has done some of these searches. Actually, this intel says we're 39% the same as a banana. Because it's a living thing, and cells have some things in common, even from different species. And so I don't know if he actually measured that or that's just a generalization, but it's kind of cute. 39% the same as a banana. So
yeah, that's interesting.
So you gotta put some judgment to this stuff.
Yeah.
You can come up with a table of numbers, but you got to put some sense to it.
People, the general public, and I don't blame them, but the general public doesn't understand the context of a lot of things,
right?
Having to do with DNA and genetics and things like that. They don't understand the minutia, like because again, man, like, man, people go to work every day. They might
Right.
an accountant or a
I'm
painter.
retired. I had time for this.
They don't have
And
time.
believe me, it took a lot of time. I've done hundreds, maybe thousands of searches.
Right.
Spent hundreds, if not more, hours. And over 7 years. I mean, this is not a full-time job. I had other things to do, and even though I'm retired, I'm involved in a lot of other activities. And most people don't have the time for that. And you need a little bit of background to get you started, you know.
Yeah, you need some kind of something to get you started for sure.
Yeah, you don't have to be a geneticist, but you have to have a little bit of mathematical and scientific background.
Absolutely.
Not a whole lot, as I said, an honors chemistry student, and I had some good ones, not many, unfortunately, but they could have gone pretty far with this, with a little coaching, you know. It's not rocket science, but it is science, and that takes some study.
Well, let me ask you this question, Haskell. If let's suppose I go off into the woods tomorrow, and I chop off a finger from a Bigfoot, and I take it to a lab, and they can get DNA from it. Would it come back unknown primate, or would there be? Do you think there'd be some other like nuance to it?
Yeah.
Like,
Well, if in fact it's closer to human than chimpanzee, which I think it is, you will get some matches to human that aren't as good as they should be for a full human. They'll have too many extra mutations, but they won't have as many as a chimpanzee, I believe. And you will know that it's a primate. You will know for sure it's not a horse, a dog, a cow, a cat, or any other animal.
mhm.
Primates are very distinct. Their DNA stands out. And it's got to be a primate. We all think that because it walks and acts like a primate. I mean, that's almost a given, not quite. But so you'll find some DNA that's not human, but it's not chimp, and it's even farther from any other primate. So, you know, then what do you make of that? That's my prediction. I could be wrong, but I think it's too human-like. In fact, I think it may be a human mutant or some strain that got off with some unusual genes and got isolated and learned how to live in the woods, but didn't need modern technology, didn't learn language, because, you know, you have to learn language around people who speak it. If you're out in the woods alone and so on, you're not going to learn English.
True.
So I think it's closer to human than any other animal.
But the result of my scenario I gave you, it would definitely come back unknown primate.
Unknown primate, yeah.
And that would
and you could what you can do is you do a comparison of the DNA to some typical primates that have sequences in the database. You can do chimp, gorilla, orangutan, maybe a couple monkeys to fill in, gibbon, and the DNA of your unknown sample, and you produce, and I got a couple chapters on this too, what's called a phyllo tree. It's a tree of life, an evolutionary tree of life, and you'll see exactly where this sample falls. Was it branched off a human? Was it branched off a chimp and went the other way and then kind of converged on human, called convergent evolution, or what? And if you have a long enough sequence, it can be very, very definitive. Let me put it that way. But you need at least 300 or 400 bases of mitochondrial. I would say at least, preferably longer and preferably some nuclear. But if you don't have it, don't try to do it on 100 bases. That's not enough, because I can guarantee you that there are 100 base segments of human DNA that match a pig 100%, because they're so short.
Right.
There's segments that either are junk DNA that don't produce genes or any proteins, or they're just part of a gene that's working real well and hasn't changed that much, So you need a longer sequence. And now Sykes' study, he only used 104 bases, but he picked a region that varies quite a bit among different species. There are some, what do we call it? Some ambiguities because, for example, white-tailed deer and mule deer come out the same in this short region because they're related. I found a relationship between a sheep and a what's called a Himalayan tar, which is a member of the sheep family, that matched exactly, so you don't know which it is. And there was one other thing, but definitely the black bear stood out as different from any other bear, as a matter of fact, or any other animal. But I would recommend a much longer sequence than 104 base pairs. I mean, he picked a really good region, and it worked because it showed, even though you don't know the difference between a sheep and a tar, who cares? Neither of them are a primate, or even close to it, right?
Yeah.
Or two deer. It doesn't matter to me which deer it is. They're deer. They're not primates. Okay. So.
Agreed.
Yeah, I was impressed with his paper. There's criticism, this and that. He cherry picked samples. I don't know. He got 58 samples, 37 or 38 of them seemed like likely candidates, and 8 of them did not sequence, did not amplify, and he got 30 results. And the one that's most of interest, the sample 26, the Justin Smasia sample, turned out to be a black bear, no doubt about it. And there was a human sample, but he realized that the 104, like I was telling you, may not be enough bases to tell the difference between a human and a Bigfoot. So he did a much greater sequence of, I think it was the hypervariable region of the mitochondrial DNA, 300 or 400 base pairs, and it matched human 100%. It matched the human standard, as a matter of fact. So it's definitely human, and unless Bigfoot is very, very human, it's not a Bigfoot. So I thought it was a good study. You can criticize the sample. He welcomed anybody to submit a sample, and some of them are still available. Portions he didn't use at the Museum of Natural History, Lausanne, Switzerland, I think. And so they can be reanalyzed. You don't believe it?
Yeah.
I think it was a good paper. Now, people say, well, he picked samples he knew weren't Bigfoot just to show that it doesn't exist.
No,
I don't believe that.
That way to me.
These are samples that well-known people had kept for years, thinking they were Bigfoot. Some of them, I think, came from Tibet. There's been some of these that look like skulls, but actually it turned out to be yak hair and that sort of thing.
Yeah.
I think that's where one of his samples came from, at least. And these are another anomaly is they found one was a raccoon, and it came from Russia, which has no raccoons out in wild, or unless somebody released some. So that obviously was either somebody's attempt to fool him, or mislabeled sample, or something, or somebody mixed some samples up somewhere along the line. But a raccoon should not be found in Russia. So that was an anomaly.
Yeah.
I thought that was kind of interesting. So that I think it was a good study. It shows what can be done, and you need the right sample, of course, obviously.
Definitely.
And I've read a number of his books. They're very interesting DNA books about the origin of human haplogroups and that mitochondrial DNA, about the DNA of the British Isles, and one interesting point, there's a lot of Scandinavian influence when they raided, and Iceland, here, get this, 2/3 of the men have Scandinavian origin in Iceland, but 2/3 of the women have English or other European origin. So the Scandinavian raiders were taking other women with them when they raided towns in Ireland, Scotland, and England. Very different. You'd expect them to be almost the same.
I thought maybe
the men are more Scandinavian and the women aren't.
Yeah, I thought.
Just a little tidbit.
Yeah, this question right here, I do believe you kind of covered it.
I was wondering, what was the most
Yeah,
bad
this question right here, somebody else wanted you to.
Yeah, I think I tried on
Yeah.
that.
Pretty
They
much.
searched the wrong databases. They didn't search enough databases, and they were too quick to settle for a 95% match, and in some cases too quick to give up when a lot of hits were all about the same in different animals. And obviously the one that really matched was not in that database, or not at the right region of the DNA. So those are the things. And I'll just say,
So yeah, basically,
she didn't get the best team together. The microscopist is pretty good.
right.
But the rest of them, they're all these forensic labs. They don't deal with wildlife DNA. They don't know how to identify new species. Right. They deal with human samples that they took out of somebody's vein or hair or something, They weren't qualified to do what they were trying to do. And unfortunately, she wasn't smart enough to realize that.
Yeah.
Right.
I feel like she kind of took the cheap route. She gathered, it was probably her cheapest route to go. And, and
I'm not even sure of
with
that.
that team that you just said, yeah, they were.
She had no real good academic connections with people that really know a lot about DNA. These little contract labs that do forensic stuff, they are not experts. They're not really even geneticists. They're lab technicians. They have a specific protocol they have to follow for it to be acceptable in court. And if your sample isn't human, you're going to get weird results, and that's what happened. Sorry.
Basically, yeah, and it all boils down to incomplete sequences, basically, on her part, and where a lot of people fail to understand that after everything that's.
She tried to sequence the whole chromosome 11, which is just one chromosome. I don't know why she picked that one, but she only got a fraction of that. The biggest fraction was sample 26, which was about 2% of the chromosome. And the other one, sample 31, was less than 1% of the whole chromosome. So if she had a reference sequence that was human, which she did, and she didn't get any better match than this, that's a good indication that the sample is not human or human-like, or even a primate. She got genes that were conserved with these other animals like dog and bear, and that's all she got. And she made it very difficult to sort through it because they were conserved genes and they didn't show as much difference as some others might have. So what are the things learned from the genes regarding?
All right.
What do we learn? Well, if you could get a nuclear DNA sequence with a lot of nuclear genes, you could really start to understand this animal. Like, why is its eyesight supposedly so good at night? Why does it see infrared? What kind of hearing does it have? What kind of smell does it have? Why does it have, what's the deal with the hair? Are there genes that produce that kind of heavy hair compared to a human? You might learn some things about its locomotion. The DNA eventually is transcribed to RNA and then translated into proteins, which are the body building blocks of the body, in the bones, the muscles, the teeth, the organs, and also the enzymes that make the chemical reactions work in the body. They're proteins as well. So if you have the DNA, you can figure out what the proteins are and how they work and why they're different from human and what that implies for the animal's behavior and physical characteristics and all kinds of good stuff. I mean, this is medical research now, trying to understand diseases through DNA. How are people with a disease different? And what does that imply for the protein that particular genes end up producing? And so, everything you want to know about an animal is in its DNA if you can get the full sequence of the nuclear and the mitochondrial. So the sky's the limit on this. They're still working on the human one. They're finding new genes and new causes for disease and all that. That's still ongoing, and they've had the nuclear sequence now for, I think, is it 2 decades? About 2 decades, 20 years or so. So a good question. Bigfoot, Okanagan. I've been to Okanagan Valley and the lake there. I did not see Ogopogo, tell him. That is the Nessie equivalent there in Alberta and Lake Okanagan that people say they see. And I missed it, but it's a great area of Canada people aren't aware of. It's kind of like Canada's only desert where the humid air from the Pacific goes up and deposits in the mountains, the Cascades, and then come over and it's very dry. So it's like a little desert. They grow grapes and fruit, and it's really more like Arizona or Israel, something like that. It's a really cool place. And I love ice wine. I never had ice wine. They pick the grapes after the freeze, and it concentrates the alcohol, and they throw the ice away and keep that concentrated alcohol thing. It's like a liqueur. Boy, it's expensive, but it's great. Wonder if all the women that get taken get their eggs mixed with the Bigfoot. Oh, wow. Yeah, that's another theory. That,
I was wondering how you would take
this. The
this.
whole thing is based on harvesting human eggs and UFOs and extraterrestrials, and that somehow Bigfoot is part of this conspiracy and all that. Yeah. I certainly have nothing to add to that discussion. I've never seen it happen. I do believe that.
I was going to ask you.
Yep.
Yeah, I was going to ask you before I put it up there if you.
Some of these UFO pictures.
Yeah, I'm sorry. You go ahead. I was just going to.
I don't know much about UFOs. My wife is a UFO fan and an Ancient Alien fan. And there's a beautiful cartoon.
Oh, cool.
On the internet. There's a Bigfoot peeking out from behind a tree, and an extraterrestrial peeking out, looking at him from another tree, and the caption is a sighting.
I think I've seen that one. Yeah.
You don't need to think that hard. But it's kind of cute. So we kid each other.
Oh yeah.
She's not sure about Bigfoot. I'm not sure about
I come across a lot of Bigfoot.
But it's fine.
Yeah,
it's
there's a lot of silly Bigfoot cartoons.
Oh yeah, it's harmless, and But anyways.
Well, Haskell, I'll tell you what. We'll go ahead and get ready to wrap this up. I definitely want to have you on very soon again for a part 2. As far as a part 2, I'll be able to gather some more information after I start reading and looking through your book. And I highly recommend everybody you can find his book on Amazon. Also on YouTube, under the description of this live feed, if you look at the description, I did post a link for his book, uh, "The Sasquatch Genome Project: A Failed DNA Study," forwarded by Dr. Jeff Meldrum. Um, so by Dr. Haskell Hart. So yeah, take advantage of it. I would go ahead and order it and get it as soon as possible. And maybe we could all have more of an insight when we
Yeah.
have Dr. Haskell Hart back on with us. And I'll definitely discuss that with you through Facebook Messenger because I do have another
Okay.
question. I'll actually ask you after we go off air here. So yeah, when we
Okay.
go off air, just stay put where you're at, and because usually we have a few words after we go off air.
Sure.
So,
Well,
but
listen, I
meanwhile,
think,
for those who are watching, thank you. Oh yeah, go ahead. You're more than
yeah, for
welcome.
the
Go ahead.
next session, let's get some people who've read the book and have questions or challenges. I think that would make a totally different program. This was a good one. I like the questions. I like your narrative. But I think to make it different, I think next time let's see what people think of what they read.
Well, if you
Absolutely.
like, if
Give
you
us some time though. This book is still new.
yeah, if you would like to be challenged, Haskell, then I mean we can get Scott Carpenter on here. I mean,
Yeah. Well, that's fine.
yeah.
I'll discuss with him or anybody, but just some other informed people are fine too. Whatever you want. It's your program, and I'm sure going to accept an invitation. I'm not afraid of any of these people, and
Good for you.
the reverse may or may not be true, but hopefully not. I do want to talk to people. I want to hear what they think. And is this book intelligible? Even I thought it was, but I may be way off base. So whatever you can do, I'll join you. And thank you. Thank you for inviting me. I enjoyed the discussion. I think it was very productive. And thank you for having me.
You're welcome.
Absolutely. Thank you for coming on. And again, like I said, just sit tight.
Okay, will
And
do.
Pat, what we normally do, we usually go around the panel. Pat, you have anything else you want to throw out there, Pat, before we go off air?
Yeah, no, it was a great discussion, and it's definitely a nice change to have somebody from the scientific community on and talk about it, so it's not just a bunch of us speculating to no end about what's going on. Just to go in a direction of actual observational science and just try and figure out what this phenomenon is, and a good starting point I think is some kind of undiscovered primate or hominin, if you will. So thanks for coming on. Appreciate your time tonight.
Sure. Anytime.
Yeah, we'll see. We'll have you back on again for sure.
Okay, look forward to it. Thank you.
Yeah, because absolutely. Yeah, because for part two, there's a couple things I wrote down. I'll say for part two, a couple more of a question and a statement, which I'll share on part two when we get you back on here again. And then for everybody else, if you're new to watching this YouTube channel, please feel free to subscribe. If you're watching from Facebook, from our ECBRO fan page, thank you. And definitely share the live stream around. And until Facebook gets the bug fixed, when you share this around, make sure you guys got to. Unfortunately, it doesn't do autoplay, so you got to click to watch the screen. But it's we're actually currently live right now. So, but with that being said, be on the lookout for part two. I'll probably title it "The Haskell Conclusion Behind the Sasquatch Genome Project." So, ladies and gentlemen, until next time, thank you guys very